Hello, I'm Doug Montgomery, and I'm glad to have you back for our November Fireside Chat. Some of you may be new to either to the course on Coursera or to this Fireside Chat. Basically, when we put this course together, we wanted to have an interactive component to it to sort of connect the course material and our participants. And so we hit upon this idea of a monthly fireside chat with a guest speaker, and the guest speaker always gives an interesting presentation about some specific experimental design topic or some application, and then we leave some time at the end for questions and answers. And if you have questions, this is a great time to put them out there. You can ask a question about anything in the Coursera course. You can ask a question about any situation or issue that you're encountering or dealing with, or of course anything about our guest's presentation. Please feel free to ask these. We ask you to put those questions into the chat, though, because that will help us facilitate how we get them answered. For those of you that are on Coursera, we put these fireside chat recordings in the course, and you can find them in the resources tab of your Coursera course. Quick reminder about how we try to run this. Please continue to keep your microphones muted, your cameras off during the session to avoid any kind of crazy background noise or things that might be a distraction. And as I mentioned, if you want to submit a question to either me or to our guest, then please do that using the chat function, and you don't need to wait until the Q&A begins to do that. You can start putting those questions in anytime you want. If we don't get your question answered during the time of the fireside chat, we will deal with those offline and will respond to those in Coursera for people that are enrolled in the course. So our guest today is Dr. Chris Gotwalt, and he leads the statistical software development testing teams for JMP Statistical Discovery. He is very interested in developing new technologies that accelerate innovation in industry and science. Chris joined JMP when he was a PhD student back in 2001. He has contributed a lot of numerical algorithms and new statistical techniques. He's authored algorithms at JMP for fitting neural networks, linear mixed models, optimal design and experiments, analytical methods for text analysis, and algorithms for fitting structural equation models. He's a principal investigator for self-validated and symbol models, a procedure that makes machine learning possible for small data sets often encountered in industry. He holds adjunct professor positions at North Carolina State, University of Nebraska, and University of New Hampshire, and he was the 2020 chair of the Quality and Productivity section of the ASA. I've known Chris really since he joined JMP, and I'm really pleased to have him talk with us today, and he's going to give a presentation on functional data analysis methods in experimental design. And this is a really interesting topic. I am really thrilled for him to do this, and I think I told him at the start of our discussions about this fireside chat that I actually encountered a problem involving this back in the 1970s. Okay, and I didn't know what to do. So I wish I knew then what I know now. But the problem that I encountered was really kind of interesting. This company that I did consulting work for was trying to develop a product that would be a competitor to Gatorade. And they also wanted this product to have some capability to rehydrate surgical patients that had been become very dehydrated as a result of the surgical procedures they had undergone. And so we had this experiment where we were taking these subjects and we were dehydrating them. And we had a particularly brutal, nasty method for doing that. We ran them on a treadmill in a humidity and temperature controlled chamber until they were physically exhausted. And then we drew blood from these subjects and analyzed the electrolytes in the blood. And what we were interested in is how fast did the electrolytes recover for different treatments that we applied. And so what we had as our response was a curve. It was a curve. And obviously you want the recovery to be fast. So how did we measure this? Well, I had no idea. So I came up with this crazy idea that, well, let's see how long it takes to get to the peak. Okay. And then let's see what the peak electrolyte level at the maximum is. And so I had two responses. And I treated them as scalar responses, which I'm sure Chris is laughing now over what I did, but it was the best I could do at the time. Confronted with a problem, you try to invent something that at least seems reasonable. So Chris, with that background, I will turn this over to you and let you talk about the really cool way that we can solve these kind of problems today.
Well, thank you very much, Doug. I really appreciate the invitation here and the introduction here today. Yeah, my first example I'm going to go through is very similar to the one that you described, so it'll be again kind of fun to make the connections there to what you were doing as the state of the art some time ago. All right, so now I'm going to share my screen and begin my presentation. So in the last couple of years, there's been a tremendous interest in the new functional data analysis capabilities of JMP Pro. There are many kinds of functional data out there under many names. We've made it a lot easier to solve problems in industry in a way that is easy to interpret and learn and leads to more accurate results. So we find that this accelerates innovation in highly competitive industries that need to develop new products and processes quickly.
We don't see your screen yet.
No? Oh, I got to click the share button. I always do this. Sorry. Fact of life is I bounce back and forth between MS Teams and Zoom, and I get acclimatized to one. Anyway, thank you.
Got it.
That's good.
So the platform in JMP Pro that supports this is called the Functional Data Explorer. And I want to say that I've been in JMP R&D now for over 20 years, and the Functional Data Explorer that I'll be showing today is probably the most exciting new analytical feature that I've ever been a part of. I need to give full credit to Ryan Parker, the principal developer for Functional Data Explorer, and Clay Barker, who has also made a lot of important contributions to the model fitting tools in there. The approach to data analysis that I'll be talking about is probably new to you today. There are not, to my knowledge, any other products that do what JMP Pro does with curve or spectral data in the way that it connects DOEs and machine learning models to curve linear data. We have several patents already in place with this regards to our very unique approach. So functional data analysis is very useful whenever your data arise as a set of coordinates or sequential measurements. The word functional is a generic term that captures many kinds of spectral and time series data. So time and wavelength are probably the most common examples of X variables, but there are many different kinds of functional data out there. Throughout the presentation, I'm going to be using words like function, curve, and spectra pretty much interchangeably. And so now I'm going to go into a couple of examples introducing what functional data is and different types. So what we see here is a plot of some functional data. So this is the home price index in New Jersey from 1990 to 2021. Like nearly all functional data, this data didn't come to us as a smooth curve like we see here, but instead as a set of discrete index values for each year. In functional data analysis, it isn't typically just one point of the curve that we're interested in. That would still be an application of like scalar DOE analysis, or even a collection of points from a single curve. We generally want to understand and model the variation we see in a set of different curves. So this is what you see here with annual home prices for all 50 states over the course of the set of years we see. Generally speaking, we're working with sets of curves that all have a distinct beginning and an end point. the starts and finishes don't have to perfectly line up, but I want to say at the outset that functional data analysis is not really used for analyze a single long-running series. That would still be the domain of tools like signal processing and time series analysis. So functional data analysis uses all the information contained in the curves by first converting the discrete numbers into continuous functions and then decomposes the variation in their shapes in ways that the functions can be used as responses in a DOE model as predictors into a machine learning model. So now I'm going to going through a couple of examples of types of curves that I've seen to get you, hopefully this will help make you see some connections to data that you see on a regular basis. So what you see here are a set of curves from a designed experiment where a company that made an ink product wanted to control a clarity measure across a range of temperatures. Here's another example. This is a set of infrared spectra of gasoline. This was a study that was done in order to see if they could come up with an inexpensive infrared based method for estimating the amount of octane in gasoline so that the inspectors could do their verification that the octane levels at stations were what they're being advertised as without having a high cost of sending the data off to a lab. Microbial growth curves are a common type of functional data analysis in the biotech industry. Raman spectra are another type of functional data that we've seen a lot of recently in JMP. High performance liquid chromatography is an essential analytical tool in the pharmaceutical production area. Here we see chemical reaction kinetics curves of the amount of a pharmaceutical product, its starting material, and two different kinds of impurities over time. So this was a designed experiment run in the pharmaceutical R&D department. They wanted to get as much of the product as early as possible while minimizing the amount of the impurities and remaining starting material that they needed to remove at a later step. The product impurity in the lower left corner was the most crucial compound to minimize as it was the most difficult to remove downstream. Here are sensor measurements for batches for fermentation process and drug manufacturing. These are the time profiles of ethanol amount, temperature, tank level, and molasses feed. And they wanted to use these curves as inputs into a machine learning model that would predict batch yield at the end of the run. So why should we learn functional data analysis? Well, we've made it easy to interpret the results. Scientists and engineers are very proficient at reading curves and spectra, which often have a direct meaning to them. And functional data analysis also lets them see what is going in visual terms that they naturally think in. You also get more accurate results. So this approach allows you to get more out of a resource you often already have because the data are often output automatically by the equipment as curves. With functional data analysis, you're using the information in the curve efficiently rather than pulling out a single number from the curve. More efficient use of the complete data leads to more accurate results and saves time and resources. Most importantly, functional data analysis is now easy. It may sound like an advanced topic, but our goal is that functional data analysis, in particular functional design of experiments, will become an integral part of data-oriented scientists and engineers working on product and process improvement in industry. So there's lots of opportunities for incorporating functional data analysis in industry, but the one I'm going to focus on today is the one that's really attracted the most interest, especially in the biotech, pharma, chemical, and consumer products industries. So it turns out that a lot of questions asked by engineers and scientists working in industry boils down to some variation of how do I change things so that I get a particular profile? We call our answer to this question functional DOE analysis or fun DOE. We want to make this as easy as analyzing scaled experiments so that if you've taken Doug's course on design and analysis of experiments, with just a couple hours of training, you'll be able to do functional analysis of designed experiments. So there's a standard workflow in a functional DOE analysis with 5 steps. The first step is the same as in any designed experiment, where you design the experiment using skills you already have and then collect the data. Then we have with some data processing steps that need to be done with functional and spectral data. A lot of this just boils down to removing unwanted or uninteresting variation from the curves prior to the analysis. Functional data analysis operates on continuous functions, so we have to use wavelets or smoothing splines to convert the discrete measurements into continuous functions. This also has the added benefit that it also kind of smooths. Can you guys still hear me? My mic popped out.
Yep.
Yeah, we're okay, Chris.
Okay, let me, okay. So once the smoothing model is fit, JMP does a dimension reduction that is much like a principal components analysis. So this identifies the different shapes that characterize variation in the functional responses and reduces the functions down to a small number of score values that explain the most variation in the data. And then we apply standard DOE tools to these scores, just like it was a regular scalar type DOE. Now I'll go into an example. So a pharmaceutical company was making a product that needed to be milled down to between 70 and 85 nanometers to be in spec. The milling time was a bottleneck in the production process, but staying consistently within the spec region with average particle sizes neither too large or too small was actually even more important. So to optimize this production step, the company did a 4-factor DOE with percentage of milling beads, power setting on the mill, flow rate, and temperature as factors. For each combination of factors, a technician would run the product through the machine and take samples at different time points and measure the average particle size of the sample. The curves look like the graph you see here. The size tends to decrease steadily at the beginning and then kind of flattens out at the end. So to solve this problem, we're going to ultimately use a functional DOE profiler that shows how the predicted particle size curve over time changes as a result of our DOE factors. So this would be the final tool that you use after all the data wrangling and modeling steps. Ultimately, what we want to do is get the curve into the middle of the spec region as early as possible, and we want it to stay into that range for as long as possible so that the process is fast and robust. So here, once we've got our model, we can vary the amount of beads. We see that getting the amount of the bead percentage to be high gets us earlier in, get us into the spec region earlier, but it's still not quite good enough. Then we can adjust the strength factor and see how it impacts our size over time curve. And this one also looks like setting the strength setting to its highest value will get us there. But when that happens, we're getting there earlier, but we're shooting through the bottom of the spec region rather quickly. So can we solve this problem in a way that's robust in addition to fast? And when we adjust the flow rate parameter, we see that the tail of the curve stays into the middle of the spec region for a longer period of time if we minimize flow rate. And then by adjusting temperature to its lowest setting, we're kind of getting into the middle of the spec region a little bit better than we were by just minimizing flow rate and leaving temperature at the minimum, at the middle. So now we found process settings that get us into the spec region early, and we stay there for a long time. We're also getting into the middle of that region by about 12 minutes. Now I'm going to go ahead and show the details of how we got there. So first we had to collect the data and get them into a JMP data table, which we're looking at here. In the data set, we have this in what we call a stacked format, where we have a batch ID column, and the time and size coordinates are columns in the data table. And we also on the right have the 4 DOE factors. Notice that they're all constant within a batch, which greatly simplifies the analysis. If you have time varying factors, then you have a functional input and functional output DOE, which is kind of still an advanced application. To analyze this as a functional DOE, we bring up the Functional Data Explorer platform in JMP Pro. I'll be calling this FDE for short. It's just our little acronym for referring to the platform. So we're going to load size up as our Y, time is our X or longitudinal variable. We have our batch ID indicator, and then we're going to supply our DOE factors as what we call supplementary variables. So once we get into the Functional Data Explorer, we see an overlay of all the size over time data, as well as a trellis of the individual curves. Note that all the curves are not all the curves are the same length, or do we have the same number of measurements per curve? FD is pretty robust to different kinds of data structures. There's a lot of options for cleaning up data and transforming it. But in this case, this data is fairly clean, so I'm not going to have to go through any of the various data cleanup tasks. Once you get your data in good shape, then we have to choose a smoothing model. In the past, in versions 15 and 16 of JMP, I really told people to go to B-splines first. There's especially for simple functions, but in JMP 17, we've added wavelets, which are seemingly better in every single way. So now they're the first place that I go for an analysis here. They're faster, more automatic, easier to use, and more accurate. I'm really impressed with how well wavelets work on functional data. So once we fit our wavelets, once we choose wavelets, it goes through and fits a variety of wavelets models and gives us a plot of the individual data points with the wavelet model fit overlain. Here we see that the wavelet model fit is in red. So it looks like it's going through the points almost perfectly. This is a really good fit. And generally speaking, we have the option to change the wavelet model, but I never have ever switched from the default one that popped up first. So once we've chosen a basis like that, then automatically Functional Data Explorer does a functional principal components decomposition of the wavelet model fit. So here it's used a model selection algorithm to identify a mean function and 3 characteristic shapes that explain the most variation in the functions. So here we have the mean function. This is just like a pointwise average across the time points for all the batches. The first shape function appears to be kind of just a simple level set, and this explains about 88% of the functional variation in the data. We have a second shape function that seems to be paying the most attention to behavior at the end of the batch. And we have a third shape component that looks a little bit like the first one, except that its directionality is going a little bit different. It's kind of modifying behavior at the end of the trajectory. So we can interactively use actual biopredicted plots at every stage of the functional DOE analysis to do our model selection. So what we have here is the actual sizes that were observed and the ones that are predicted by the model. Here we can, it looks like this 3-component model is going through the data pretty well because it's kind of sticking tightly to the 45-degree line. But we can add more dimensions to it, just like a principal components analysis. Here we're looking at 5, and I don't see a big difference between 3 and 5 principal components here. So I don't necessarily think that the 5 principal components are worth it. 4 also doesn't look particularly different. Here's 3. We go down to 2. It looks pretty close to 3. If we go down to 1, now we're starting to miss variation. We're starting to see patterns in our actual biopredicted plot. If we go down to 0, this is the same as just working with a mean function. So we definitely don't want to use that. And overall, I'm just going to go ahead and stick with the defaults, which is what I usually do, and work with the 3-component model that came out in this case by default. So the functional principal components model breaks the data into 2 kinds of variation. There's the shape functions, which characterize all of the time variation within the batches. And there are also these functional principal component scores that characterize all of the batch to batch variation. So for example, function 2887, the first batch, we're approximating that function as a function of time, the mean function, plus a functional principal component score of 112.3 times shape function 2, minus 8.1 times shape function 2, and plus 1.0 times shape function 3. That's our model that we're going to be working with. And ultimately, we're going to take these FPC scores that explain all the batch-to-batch variation and apply DOE modeling tools to them. So this would be pretty much equivalent to creating a data table with one row per batch and our DOE factor settings here as columns, as well as our functional principal component scores, going into JMP's fit model platform, and then separately modeling the functional principal components. Fortunately, we've made it a lot easier in the Functional Data Explorer by automating this process with a functional DOE analysis. So we click this and it fits models to the FPCs using best subsets regression, and puts together this FDE DOE profiler that connects our factors to the predicted response trajectory. And just like we did before, we can kind of piddle around with our factors and ultimately, find a combination of settings that solves our problem directly. Now we could just go straight to that profiler. We do have the ability to open up some generalized regression platform tabs for each of the FPC scores, where we can see what particular factors and interactions are predicting those FPCs. There's some interpretive value there, and you can go through and customize the models and add and remove terms as your kind of experience dictates, or or if you like this, what you see with the results better that way. And that's my first example. So
this is really amazing. I just have to jump in and tell you what an amazing tool this is, because I don't know whether people really appreciate this, but there's a lot of very complex machinery going on here under the dashboard. So I'm really impressed with how smoothly this works.
Yeah, it is. I can talk about it in great length, but there's a lot sitting on this. Yeah, so it's been really remarkable to put so much machinery into a user interface that solves such important problems so easily. So I'm going to go over a second example. So, often the goal of a functional DOE analysis is to find a combination of factor settings that allow us to realize as closely as possible an ideal trajectory that was defined in advance. So we call this golden curve analysis. And for my example here, I'm going to show how you do this. This was motivated by a JMP customer that supplied chemicals to a consumer products company that made dishwasher soap. The consumer products company needed their soap to maintain a specific surface tension over time curve, so that at the end of the dishwasher run, you would try to minimize spotting on your glasses with this. So the consumer products company sent to our customer this curve, which represented what they wanted the surface tension to look like over time. And the chemical products job was to find a formulation that would match this curve as closely as possible. So to do this, they did a 3-factor mixture experiment. Here's a trellis plot of the design. The analysis is very, very similar to the first one, in that we're analyzing a data set that's in a stacked format. But there's this added little piece where at the end of the data table, they joined the XY coordinates of the ideal curve, so we can match that in the analysis. So now I'm going to do a live demo of this. So here is the data table. I'm going to go into the Functional Data Explorer. And I'm going to use ID as my batch ID. The 3 factors are my explanatory variables. Time is my X coordinate and surface tension is my response function. So now we're in the Data Explorer. So we've got, I'm just going to call out that we have all these great tools for cleaning up functional data. We can transform the responses and also the inputs or the longitudinal variable. We have a bunch of different alignment options, so you get the the X's kind of lined up for the functions. In JMP 17, we've added a lot of spectral analysis tools like multiplicative scatter corrections, standard normal variate transformations, Savitsky-Golay smoothers, and derivative filters. I'm actually going to undo that step. And we can also do baseline correction. But for this problem, because it's a golden curve analysis, what I'm going to do is I'm going to go to the target functions and I'm going to load up the target function by selecting its ID value. So now it's loaded that up. And that's all the data processing I'm going to do. So I'm going to move on to my wavelets model. Wavelets require an evenly spaced grid, so it's kind of doing that automatically for us in the background. Okay, that's my screen here. So here's our functional principal components analysis. It seems like wavelets tends to over-select. We get lots of we have lots more functional principal components than we really need. Kind of the good news is that with functional DOE applications, it doesn't really hurt to have too many. So like if we mess up the DOE model and shape function 4, for example, we're only kind of messing up 0.1 of the variation in the model. So like overfit with this side of things is not really a problem. And so from there, I can bring up my functional DOE analysis. And here's our functional DOE profiler. And because this is a golden curve analysis, we have some options for working with the target. So the initial profiler here is our surface tension over time curve. That is a function of our DOE factors. You can see how that changes. Now we can click this button, and this brings up 2 more rows in this profiler. One is a pointwise difference of the predicted trajectory from the ideal curve. And this lower one is the integrated squared distance of the predicted curve from the ideal curve. And this is what we want to minimize. We want to get as close as possible. So we just click optimize target. And well, it's not. Oh, there, it just took a second for it to sync. So what it's found for us is that we can get as close as possible to the ideal trajectory with a formulation of 57% of factor A, 36% of factor B, and 7% of factor C. So you could do some test runs, confirm that the formulation is working the way that the model predicted, and then that would be the product that you send off to your customer. So we're at 7 after the hour here. Should we take some questions, or I can go into other examples that are spectroscopic oriented?
Well, I'm really very interested in this last example that you just talked about, because I myself have seen similar problems to this in consumer products, because very often you are trying to either formulate a product or produce a product where there is a customer expectation as to what this thing is going to look like, how it's going to work, and being able to match that profile, if you will, is a really critical part of either the product development process or the or the getting the manufacturing process working correctly. So this is really a kind of a remarkable thing that you can do. And I guess the next question I have is have you published anything on this outside of JMP? You should, you could take this example and get a really good article out of this in something, some journal that would give it wide publicity, and it would really encourage a lot of people to learn more about this technique.
I agree. So I've not done really enough to get a publication out there on this. I tend to, I've got a meeting tomorrow actually with some folks I'll be collaborating with to put together a series of blog articles and possibly some papers on this, because I think this really needs to kind of get out there. But I generally am very busy with JMP R&D stuff, so I often get, or we often get behind in publishing the things that we put in there.
I understand, and as I'm an editor of a journal, Quality and Reliability Engineering International. If you guys write something up, send it. I'd love to have you send it to our journal, because we have a big circulation, international circulation. We have a big impact factor. I'd like to see this methodology get more into the mainstream of experimental design practice.
Yeah, well, I have a meeting tomorrow to talk about this very thing. So, and
Good.
we will, QREI is our likely target for talking about this. So you'll hopefully see something from us here in the next couple of months.
Yeah, and the next time I revise my experimental design book, I'm going to have to include material on this, because
That's wonderful.
This is something that really needs to get into practice pretty quickly, because this is these problems show up a lot. Yeah. I told you about the athletic replacement fluid problem that I got involved in the 70s, and I'm embarrassed at what a crude approach I used to attack the problem. But since then, I've encountered this kind of problem over and over and over again, and not only in the experimental design world, but in the process control, process monitoring area. I remember doing some work some years ago with a student was involved in this, where the company manufactured airbags. And the principal quality characteristic in an airbag is the inflation curve, of pressure versus time. And there's a set of specifications that you have to match on that, because if it inflates too slowly, it doesn't work, and if it inflates too fast, it injures the driver, so or the passenger. So in their process monitoring scheme, they would test these bags and look at the profile. So how do you monitor that from a statistical viewpoint? That's again another problem connected with. Yeah,
with I've seen you have. I saw your publication on that. I need to take a closer look at it. There's these problems are everywhere. you can turn pretty much like a lot of industrial statistics. You can take any topic in there and kind of insert a functional at the beginning of it, and you've got a whole green field of new areas that you can research. Yeah. I had also one of the first applications we'd seen where people wanted to analyze functional design experiments was it was an automotive supplier. They make safety belts, and every automotive company, Toyota, Mercedes, Ford, when they were purchasing seat belts, they had a specific force versus distance curve that characterized what they wanted their passengers to experience when they're using their seat belts. And the company had really struggled for a long time on this, but they'd obviously been successful, but they were really looking for a more direct way of handling this, and so they jumped as soon as they saw functional data analysis. they were particularly interested in also being able to do a measurement systems analysis of functional data. So Laura Lancaster, my colleague, and Astrid Gluck, the customer that we were working with, we have a white paper on this up on the JMP website. Just a very simple approach for measurement systems analysis. Control charts, SPC for this is wide open. There's functional machine learning where you try to take curves from multiple sensor streams and use that as inputs into like a neural network to predict batch yield or other kinds of critical quality attributes. There's what would a capability analysis mean for a set of curves that are defined by a functional spec? I don't know yet, but I think that those are things that we should see developed here in the next coming years. So
how much of what you have demonstrated here is in, I think JMP 16 is what I have on my computer. Is this all in 16, or is this some of it?
The only thing that I haven't that is not in JMP 16 is so in the models I've been using wavelets today.
Yeah.
But in the past, this is a new addition to JMP 17.
Okay.
We also have B splines and P splines. Those were kind of like the main tools I used for this in the past. And you can, I actually just spent the last couple of days kind of updating my slides to remove all the B spline and P spline references and convert them to wavelets, because I find that the wavelets just get the model right out of the box faster and easier than the spline models do. But that would be the only difference between the version of JMP that you have and JMP 17. And
17 is out now, correct?
Yeah, like just as of the other day, actually.
Yeah, and we will have it very soon. I just need to figure out when it shows up at ASU and download it to my computers as well. So that's great. That's great. This is really terrific. Really terrific.
Thank you.
I wonder, do we have any participant questions or any audience questions that we need to deal with?
Yeah, so there's a few Q&A questions. Chris, you should be able to see them in the Q&A portion.
Right.
I can't
share with sharing your screen.
Yeah, I have to. I'm sorry, if I stop sharing my screen. Okay.
Yeah, that might be good.
Oh, okay, there we go. Now I have it's a little easier for me to read. So we got the Q&A tab here. So hello, the supplementaries are constant. So when do you change them? Well, so the supplementary variables are constant within a batch. So let me, I'm going to reshare real quick, and I have to make sure I click the share button. So let me just go back to this, the surface tension curve. Clean up my desktop. So here the factors are constant. The supplementaries are constant within a batch ID, but when you go to a different batch, then they change. So what happened initially was this was a designed experiment that was created in JMP that had 19 runs, and that created a table with just 19 rows. And when the experiment was executed, they collected the surface tension over time curves, and they had to do a join operation to blend the DOE factors table to the curves in order to get something that was in a readily analyzable form. And let's see.
And
So,
Megan's loading up these the questions. We have several. So
one question is like, is there a situation where you'd advise against using functional data analysis methodology? Well, I would say that one place where I've seen people get a little bit lost here is when they have one function, like they'll have one manufacturing process and they're collecting one measurement over time So they really just have one long time series and
Mhm.
that's it. So there they start thinking about functional data analysis, but that's really still a time series application or like a time series or a signal processing type application. You got to have like multiple instances of the functions, and they need to have well-defined beginnings and end points that kind of line up somehow in order for this to really make sense.
And that's what you would get in a typical designed experiment, because you'd have different combinations of factor levels, and each of these different combinations of factor levels would produce one of these curves.
That's right.
Yeah,
that's right.
Yeah, when you've just got an operating process with an output, it's a time series problem, and it could even be a fairly straightforward SPC type problem.
Mhm.
It just depends on the context of the problem. But
Mhm.
but as I say, I think there are a lot of situations with designed experiments where this type of data occurs. So it should be pretty obvious to a lot of people when you've got this situation.
The other situation at the other end is where you have, if you only really have 2 measurements per batch, that's kind of too small.
Yeah.
You can fit, if you want to do something, you can fit lines and slopes to each of those and pull those out as features. But like splines and wavelets are kind of overkill for when you have very few measurements per function. I'd say 3 is kind of the minimum. 4 or 5 or more is best.
But a lot of these problems produce a fairly large number of
yes. A lot of the time it's just the output. A lot of the time the equipment itself naturally exports functions already.
Right.
And so what people have been doing, like you were doing in the 1970s, was taking these big collections of data that came off of test equipment, doing kind of hand feature extraction, kind of thinking, okay, what's important here, pulling those essential features out, and then doing DOE modeling of those kind of manually curated responses.
Yep.
And so like now that data is just sitting there, and we've given you a tool now that lets you make most efficient use of that possible.
Yeah, you're not throwing away 90% of the information in the experiment, which is to me the real power of this.
Right. And I think that having the functional DOE profiler there at the end is the engineers and scientists, they'll understand what this trajectory is and what it means for them in a way that's more direct than pulling out the crossing time and then doing a crossing time model and optimizing that on the one hand is not terribly direct, and on the other hand, you may be focusing on the crossing time, but as we saw in that first example, doing a simple minimization of that at the outset caused other bad things to happen with the predicted trajectories. Like the curve was shooting through the floor of the spec range there at some of those combinations of factor settings.
Much too quickly.
And this prevents you from doing that. You won't be over optimizing on just one thing when you actually might actually care about more.
And I think it's really important that to note that the profiler works the same way it does in pretty much a standard design of experiments. So there's really nothing new to figure out. There's nothing new to really learn if you're used to using the prediction profiler, which I think most people that use JMP for designed experiments do rely on the prediction profiler. And we use JMP throughout our Coursera course. So people should in our course should have had some good exposure to the profiler. And
yeah, we're trying very hard to make it so that we're just building on tools that people are already familiar with. We want the delta from scalar DOE analysis to functional DOE analysis to be as small as technologically possible. And every release, we're kind of shortening the steps. I could talk at great length about how difficult this was in jump 14, and then we made it easier in 15. And it's been a process of peeling away difficult steps and simplifying them out of the analysis while still providing the opportunity to go in and customize things like.
Well, it's this to me, this is one of the major steps forward in the analysis of designed experiments over the last decade. This is really a giant step forward for the field, and I've really am pleased to see that it's been integrated so nicely into software. We had one or two other questions, I think.
Yeah, there was one. There was one. I should have probably addressed this one earlier. They were asking what I meant by the golden curve analysis. So what I had meant by that was that we had an ideal trajectory, a curve that was defined in advance, and we wanted to find the factor settings that would give us a predicted curve that matched that as closely as possible.
And this kind of problem occurs a lot. I've seen this in consumer products areas where as a result of maybe doing a focus group, you have customers that have said, well, this is what we want this thing to look like. This is the behavior that we're looking for. And so now what you have to do is go out and figure out how to either formulate your product or manufacture your product so that it matches that curve.
Yeah.
And this is a pretty common situation. And up until what you showed in that example that you presented, people would do this very heuristically. There would be a lot of trial and error involved. I mean, they would typically, with a designed experiment, they would figure out what factors were the ones that influence the performance. But then finding the combinations of those factors that produced that curve was almost a trial and error type of process, often done in a small scale in a development environment. And then, of course, you have the problem of scaling that up to a full scale process. And what you did with that example makes that whole activity so much more transparent.
Yeah, we're at Jump. Our kind of mission number one is in accelerating the pace of product and process innovation. And I think this functional data explorer tool that Ryan Parker and Clay Barker have created is
it's pretty nice.
Powerful.
The other application of golden curve analysis that I've seen is kind of funny is you're trying to match the competitor's product.
Yeah, that's true. That's true.
That's right. You have a competitor that's one of the market leaders, and you'd like to be able to match the performance of their product, and that's another place where this golden curve analysis comes into
absolutely
into play.
Stability curve analysis also, or if you're producing biosimilars and trying to create a generic version of or an alternative version of an existing drug. There's all kinds of things like the stability curves, the dissolution curves.
Right.
There's all kinds of data that is inherently functional in nature that would benefit from this.
Right. I've seen similar problems working with people in the pharmaceutical industry, where you look at things like a drug absorption rate. How fast does this stuff absorb into the bloodstream, or tablet dissolution? How long does it take this tablet to dissolve as a function of the binders and other kinds of ingredients that go in the tablet? And so here's one more question, which is kind of interesting. Would there be a difference if the wanted diagram is a straight line to the x-axis?
Not at all. So that initial MIL-DOE example, if I had given it an ideal curve or a golden curve that was, well, if you remember, the goal there was to get within the spec range of 70 to 85 nanometers as quick as possible and to stay there as long as possible. So I could have, so what is the what's the number that's between 7085. So that's like 77.5. If
Right.
I used an ideal curve that was 77.5 throughout and solve this as a golden curve analysis, then that would work just the same as it did with the surface tension.
So that's just kind of another way to formulate the
Yes.
the objective. Yeah. Okay. All right.
Yeah, I was just trying to I didn't want to, there is a lot here and I wanted to kind of introduce things in a step-by-step way, you know. But yes, you could solve, that's the simplest kind of golden curve analysis.
Great. Yeah, I think there's the applications of this golden curve analysis are there are many applications. I in my own experience, I've seen quite a number of them. So this is a big, in my view, a real important innovation. And I hope the people that watch this fireside chat really appreciate what a tremendous innovation in the analysis of designed experiments
Thank you so much.
You've shown us. Okay, so it looks like we are getting to the end of today's fireside chat. We do these every month. We are not going to do one in December. It's the end of the academic semester, and trying to squeeze this into everything else that's going on is beyond what I can handle. So we're not going to do one in December, but we will be back in January, and so we will have a full slate of these for next year. Franklin D. Roosevelt did fireside chats every year from 1933 until he died. You know, that was a long time. I don't know that I'm going to be at them that long, but we've got another year of them all lined up. So we're looking forward to seeing people in January. And again, Chris, thank you very much for this really important and I think innovative presentation.
Well, thank you so much for the invitation. This has been a lot of fun, and I really appreciate having the chance to be able to talk about this. I think that there's just a tremendous impact on the world that this tool is going to have for industries.
Well, and if you run across any interesting examples, let me know, and maybe we can come back and have a whole session of examples in a fireside chat, because
I would be happy to do that.
I think the more people learn about this and get exposed to it, the better.
Yeah, the important thing that I wasn't able to get to much today is spectral analysis. So, analysis of data where you have many peaks, and this wavelet capability that was added in JMP Pro 17 opens the door to that. And that's going to be one of the primary things that we're going to be working on in the future, doing things like, finding areas under the curve as a way of measuring impurities or products in chemical and pharmaceutical applications. That kind of peaked data is all over the place, and that's where I see the biggest opportunity.
And wavelets would handle that much better than splines.
Oh yeah, it's night and day.
Okay, I guess we'd better shut it down.
Yeah.
And thanks again, and hope everybody has a good holiday season.
You too. Thank you so much. Take care.
Bye-bye.